All right, welcome to chapter 11, which is all about enzyme catalysis. So in this chapter, we're going to split it into two main sections. The first part, which is this video, is going to be all about how to define and categorize different enzymes. So first we'll discuss the general properties of enzymes. Then we'll talk about some of the different types of reactions that enzymes can catalyze. And finally, we'll then discuss some of the mechanisms that enzymes use to help actually do what they do. And then in the next video, we'll talk more about serine proteases, which are a particular type of enzyme. So first of all, before we get too far, we should make sure we all know what are enzymes. So enzymes are biological catalysts, meaning that they help to carry out a reaction. And they are biological, meaning we see them in living systems, and they are performing catalytic functions around living organisms, helping reactions to go. Enzymes are usually made of proteins, or they can also be made from RNA, and there are some enzymes that are also a mix of proteins and RNA. And so essentially what do enzymes do is they make reactions happen. And so they make reactions happen faster, meaning they change the rate. They also make reactions happen under milder conditions than they otherwise would if there was no enzyme. Meaning in a biological environment where the pH is pretty close to neutral and you're in an aqueous solution, right? They also help a reaction happen with a very specific substrate that you wouldn't necessarily be able to choose if you were doing this reaction uncatalyzed. And enzymes also make reactions happen in a more regulated way than if there was not enzyme catalysis happening. But two really important points, right? First of all, enzymes do not change the overall energy or spontaneity of a reaction, meaning they're not going to change that free energy change, whether it's positive or negative, meaning that if a reaction is not spontaneous in a particular direction, an enzyme is not going to make it go. Enzymes only help reactions that are already spontaneous, help them go faster. So if you had a reversible reaction where the forward direction was non-spontaneous, but the reverse direction was spontaneous, if you added an enzyme there, that enzyme would help the reverse reaction go more quickly, but it wouldn't help the reaction go forward if it's not spontaneous. So essentially, you can think of enzymes as making a change that is in the kinetics of the reaction, in the rate, and not a thermodynamic change, so it's not changing the direction. All right, so now that we've gotten all those points down, let's think more about enzyme catalysis. So first of all, enzyme catalysis takes place in a location in the enzyme called the active site. And it is essentially responsible for the enzyme's specificity for its substrate. The active site is often essentially a little pocket, kind of like we see in this picture on the right. And that active site is a very good fit that is for a substrate, which helps it be very specific to its substrate. And there's a few different reasons for this substrate specificity. One of them is geometric complementarity, meaning that the shape of the substrate is complementary to the shape, the space in the active site of the enzyme, so they fit very well with each other shape-wise. And this, there are two different models for geometric complementarity in enzymes. One is called the lock and key model. And in the lock and key model, the substrate and the enzyme are both just exactly complementary shapes to one another, so the substrate fits into the active site of the enzyme just like a key would fit in its specific lock. The other model is called the induced fit model, which is similar to the lock and key model, except the substrate and the enzyme might be very close shapes to one another, but not perfectly exact. But when the substrate binds to the enzyme to form a complex, then they fit together more closely and interact together more tightly. So either way, both of these models are telling us that the substrate and the enzyme have to interact with each other very closely in order for that catalysis to happen. And neither model is both completely correct, nor is it either completely incorrect. There are some aspects of both models in various types of enzymes. So aside from that geometric complementarity, it's also really important for a substrate and an enzyme to have electronic complementarity. And this is where, essentially, where are the electrons going? You want them to be attracted to one another. So regions of the active site that are hydrophobic might be attracted to a nonpolar area of the substrate. A charged area on the enzyme's active site will be attracted to the oppositely charged area on a substrate. And more polar regions of the enzyme's active site will also want to interact more closely with polar regions on a substrate. So all of those types of things can contribute to the electronic complementarity of the substrate and its enzyme. And then the last thing that enzymes can do, which is pretty nifty, is that they can be stereospecific, meaning that they they're made in such a way, or they are constructed in such a way that they will interact very specifically with one particular configuration of atoms and bonds in a molecule. So let's look at an example of that. So here we have an example of a substrate. This substrate is not chiral yet, right? We have a central carbon here, and it's bonded to 3 different things, right? A yellow hydroxyl, a pink carboxyl, and 2 blue acetyl groups. But 3 very specific of those groups on that central carbon are able to bind to complementary sites in that enzyme, right? This enzyme is stereospecific. It wants to bind over here to this hydroxyl group, over here to this carboxyl group, and over here to this one specific acetyl group. This acetyl group that's shown towards the top is not going to interact with that enzyme's active site because of the arrangement and orientation of the enzyme's active site. It only wants to interact with the substrate in this very specific way that we see here. And what is this an example of? This is the enzyme aconitase. And aconitase catalyzes the reaction from citrate to isocitrate, where citrate is our original molecule that's achiral. It has this central carbon, but is bound to only 3 different things, a hydroxyl, a carboxyl, and 2 acetyl groups. And so what aconitase does is it takes this hydroxyl group onto that central carbon and moves it to one specific of those acetyl groups, that one that's shown here on the bottom, and it never moves it to the one on the top. And so it creates this stereospecificity and makes a chiral molecule out of a molecule that previously was not chiral. So something else that's really important when we're thinking about enzymes and active sites and enzyme and reaction catalysis is that some reactions that use enzymes also require cofactors. And this is often the case for reduction and oxidation reactions, as well as group transfer reactions. And so cofactors are essentially helping out the enzyme to catalyze the reaction. And so there are 2 main classes of cofactors. The first is metal ions, which we'll talk about later in these slides. And then everything that's not a metal ion can be called a coenzyme. And coenzymes themselves can be separated into 2 categories. Prosthetic groups. And prosthetic groups are always attached to an enzyme, and those are different from co-substrates. Co-substrates enter and exit the enzyme for each reaction along with the substrates of the reaction, hence the term co-substrate. So how else can we classify enzymes? We can classify them by the types of reactions that they help to catalyze. And there are 6 main types of reactions that enzymes can help to catalyze, which are shown in this table on the left here. And let's go through each of them and give a sort of example or prototype reaction for each. So the first type of reaction are oxidation reduction reactions. And these are catalyzed by enzymes called oxidoreductases. And we see on the right, number 1, the example of an oxidation and reduction reaction where we have our reactant A is getting reduced into the product B. And while A is getting reduced into B, we have this co-substrate NADH that's getting oxidized into NAD+. And so NADH is essentially donating this hydrogen, which is reducing the B, and at the same time NAD is getting oxidized. And so oxidation and reduction can be a little bit tricky or confusing to think about sometimes. In the case of these types of reactions, it's helpful to think about reduction as the gain of electrons and the oxidation as the loss of electrons. When you've learned about it previously, you might have thought of it more as maybe the oxidation state of a single atom, and you can add them up and work with them in that way. But in this case, it's helpful to think about that movement of electrons. And so when we are looking at these oxidation reduction reactions, we'll often see one of these co-substrates that we're looking at here. So the example that we saw was NAD plus and NADH, where NADH can donate that hydrogen, which has electrons, right, to the substrate to help create a reduced product, while NADH itself gets oxidized to NAD plus. There's also another pair that is NAD plus and NADPH, as well as FAD and FADH2. And the types of enzymes that catalyze these oxidation reduction reactions, you'll sometimes, when you see an enzyme that's called a dehydrogenase, you'll know that that is catalyzing a redox reaction. And you can sort of help remember this just from the name, right? So a dehydrogenase is referring almost more to the co-substrate here. D meaning removal, hydrogen meaning hydrogen, and that's what we're doing, right? We're removing this hydrogen from NADH to help catalyze this reduction reaction of A to B. So let's look at the next class of reaction that gets catalyzed. I can't talk today. And that second classification of enzymes are called transferases. And what transferases do is they transfer functional groups. And so we see that example here in reaction number 2. So X is that functional group that's getting transferred from A to B. So on the left we have our reactions. A is attached to X, and we also have B. And then the products, we have A is by itself because X has been transferred over to B. So our products are A plus BX. And what might X be? Here are a few examples. So X could be a phosphate group, it could be a hydroxyl group, it could be a methyl group. These are 3 really common groups that we see transferred from one molecule to another. So that brings us to our next classification of reaction, which are hydrolysis reactions. And these types of reactions are catalyzed by enzymes that are called hydrolases. So again, with all of these, make sure you're paying attention to the names. A lot of times they'll give you clues as to what they do. So hydrolases, they're using water to cut a molecule, right? So for our example reaction number 3, we have our reactant A, which is one larger molecule, and then we have the products B and C. So A is getting broken up into 2 pieces, B and C, and we have water as one of the reactants that's helping to do that. So that is similar but different to reaction type number 4, which is group elimination to form double bonds. This might sound very different from hydrolysis at first, but as you can see from the example reaction, we still have our reactant A, and it's getting broken up into these 2 products, B and C, but this time we don't have the addition of water. And so since we're not doing hydrolysis, we're doing this group elimination. The types of enzymes that do this are called lyases. And so 2 examples of these types of lyases are dehydratases and decarboxylases, where the group that's being removed can be water or carbon dioxide. And so, like that might be, B or C as one of the products, right? Water or carbon dioxide that's getting removed. So that brings us to reaction number 5, which is isomerization. And isomerization is carried out by enzymes called isomerases, which should not be surprising. And what this is doing is helping to, we have the same atoms in the molecule, but we might be configuring the bonds in some other way than where they started. And enzymes that carry out these types of isomerization reactions are called mutases. So that brings us to our last type of reaction that we can catalyze. And this is the formation of bonds, and that is coupled with ATP hydrolysis. And the types of enzymes that do this are called ligases. So we have almost the opposite of some of the reactions we saw earlier. Now we have two reactants, A plus B, and they get joined together to make the product C. And at the same time this bond formation is happening between A and B, we have this bond breaking happening in ATP, where we have an ATP reactant that gets cleaved into the product's ADP plus inorganic phosphate. So that phosphate bond is being broken, which is lending energy to help form the bond between A and B to help create that final product C. So now that we know the types of reactions that enzymes can help catalyze, we should start thinking about how enzymes can actually help to speed up that reaction. What are they actually doing? And essentially what a lot of them are helping to do is to stabilize the transition state. And by stabilizing the transition state, this ultimately helps to decrease the activation energy, which results overall in this logarithmic increase in the rate of the reaction. So what did those bullet points even mean? Let's look at this energy diagram to help think about that. So in this reaction, we have our reactants A and B start out at this sort of baseline level of energy shown by G on the y-axis, and we have our products P and Q. They are at a different level of energy that is at a lower G than the reactants, which is why this reaction is spontaneous, right? Because the products are lower energy than the reactants. And the overall difference in energy after this reaction is carried out is this delta G of reaction. And this is pathway independent. So no matter how you get from A and B to P plus Q, that delta G will always be the same. What changes is the other part of this energy diagram when we're talking about enzymes, right? And that's so we started with our reactants A plus B. They had to travel, before they became P and Q, the products, they had to travel through this transition state, which is shown up here at the peak of energy. So the transition state is this intermediate form that is relatively unstable and higher in energy than both the reactants and the products. And the energy change needed to get here is the activation energy, which is shown over here. And this activation energy is affected by an enzyme. Because if the enzyme is able to lower that energy state of the transition state, the difference in the delta G required to get to that better transition state is less, right? And so overall, that means the activation energy is lowered, which helps to overall increase the rate of the reaction. So let's look at what that difference would look like. So in this middle energy diagram, now we have two different reactions shown on the same diagram. One is catalyzed and one is uncatalyzed. I'm just erasing this to make some space. So, our uncatalyzed reaction that we were considering on the left is shown in blue. Our catalyzed reaction is shown in red. And so you can see that the one difference between the catalyzed and the uncatalyzed reaction is the change in the activation energy required, right? The catalyzed reaction has a much lower energy level for its transition state as compared to this uncatalyzed reaction. And so this here is the difference in energy that the catalyst helped create, right, by making that lower energy transition state. At the same time, we have this change in the activation energy, but we do not have any change in the energy level of the reactants or of the products. The only thing that changed is that activation energy. And so this middle diagram is showing what happens for any catalyzed reaction, regardless of what type of catalyst you're using. If we're thinking specifically about catalysis using enzymes, we actually would resemble a little bit more this energy diagram on the right. The curve that's shown here in blue is what we are sort of looking at. We're looking at an enzyme catalyzed reaction. So it starts out with our reactants. So here we have E for enzyme and S for substrate. So we have enzyme and substrate are separate, and then they come together to form this enzyme substrate complex, which is lower energy than when the enzyme and substrate were separate. So this is very stable in comparison. And then we have that change to this transition state where there is some activation energy, but it's much lower compared to the activation energy of our original reaction, right? And then finally, once the substrate is converted to product, we have this enzyme product complex, which is very low energy, very favorable. And then there's this very small increase in energy that's required just to separate the enzyme from the product, but that's an relatively easy energy level to overcome. And then ultimately, once everything is separate, the two different types of reactions come together again. So once again, the reactants and the products themselves do not have a change in energy. It is only that difference in activation energy that matters when it comes to an enzyme. So now that we've gone into how enzymes help to make reactions go faster, let's look at how they do that, some of the different mechanisms. And so on the left here, we have a list of a few different mechanisms that enzymes can use to help to make that more stable transition state. So that list is starting with acid-base catalysis. Then we'll go into covalent catalysis, metal ion catalysis, proximity and orientation effects, as well as preferential binding of the transition state. But first, we'll consider acid-base catalysis. And then on the right, we have some of the characteristics of acid-base catalysis. So first, we should think about what does it mean, right, to do acid or base catalysis. And for here, it helps us to think about the Bronsted-Lowry definition of acids and bases, where acids are donators of protons and bases are acceptors of protons. So for general acid catalysis, we would have a proton that's donated to a substrate. And for general base catalysis, we would have a proton that's removed from a substrate. And if we were to have concerted acid-base catalyzed reactions, both of these things, the acid catalysis and the base catalysis, would be happening simultaneously. So ultimately, to make acid-base catalysis happen, this is going to involve an enzyme's active site that will have amino acids that are able to donate or accept a proton. And this is almost always going to happen when you have amino acids in that location that have ionizable side chains, or pKrs, right? These are side chains that are able to either donate or accept a proton. And specifically, a lot of times what we'll see in acid-base catalysis active sites are histidine residues. Because if you recall from our discussions of amino acids, the pKr of the imidazole ring of histidine, that side chain, has a pK of about 6, which is pretty close to physiological pH. And so small differences in the local environment can help make that histidine pretty readily either donate or accept a proton, depending on what state it's in and what state the environment's in. So for our example of acid-base catalysis, we're going to look at an enzyme called RNase A. As you might guess from its name, it helps to cleave RNA molecules, right? And the reaction that it catalyzes is shown in this green box on the right, where our reactant is a larger RNA molecule. And then we add a water, and the products, there are 2 smaller molecules of RNA, which are labeled RNA1 and RNA2. So this is a hydrolysis reaction, right? We're adding water and we're cutting a larger molecule into 2 smaller molecules. And the way this is done is shown in this diagram on the bottom. So in the active site of RNase A, we have 2 histidines. And we were just talking about why histidines are so great in these types of acid-base reactions, because they're able to donate and accept protons, and we'll see examples of that here. But before we get started, let's just orient ourselves a little bit with our substrate, which is RNA. So RNA, as you know, is directional. You have the 5' end, which is shown here, And then the 5' end is attached to a phosphate. The 3' end is also attached to another phosphate, and then the next ribose molecule, right? And this makes up the backbone where you have alternating phosphate groups and ribose sugars, phosphate, ribose, and then on the one carbon of all the riboses, we have a nitrogenous base that's attached, and that is the basic structure of RNA. In case you needed a refresher. And now let's talk about how RNase A is able to cut this large RNA structure into two pieces. And it does this by working with the 2' hydroxyl group on ribose and the 3' phosphate group that we see. So this mechanism starts with our histidine 12. There is a lone pair on this nitrogen in the imidazole ring, which then attacks this proton on this 2' hydroxyl group. This allows for a nucleophilic attack on the phosphate group that's on this 3' carbon here. And this ultimately ends up in the histidine 119 donating a proton here. And this allows this bond to be broken between that phosphate group that we were just talking about and this next ribose sugar that's part of the next nucleotide in the RNA. And then, so what happens now is this bottom 3' piece of RNA is now not attached to anything, right? So it's able to float away, and that's what we see down here, where this bond was cut, this proton was donated from this histidine 119 right here, and now that RNA molecule can float away from the active site. But now we had from that piece of RNA on the 5' end that we saw at the top here, something kind of interesting happened, right? So when we had this nucleophilic attack on the phosphate group, the enzyme ended up actually forming this, excuse me, why does this keep zooming? This cyclic, nucleotide, where the phosphate group is attached both at the 2' and 3' ends of this ribose sugar here. So, and something else happened too, right? So this histidine 119 donated an electron, histidine 12 accepted an electron. And so the enzyme, in order to catalyze the next reaction, to cut its next piece of RNA, right, it needs to go back to where it started. And so that's actually where this water molecule finally comes in. So the water molecule comes into the active site. It's going to be able to donate a proton to histidine 119 at the bottom, and it's going to donate a hydroxyl group to this little cyclic phosphate over here, which ultimately will help to take a proton back from histidine number 12 at the top. And what we see there, so this picture is just the picture from the right in the previous slide where the water came in. It helped to donate a proton to histidine 119 and reacts with the phosphate group on top to help to take the proton back from the histidine 12 at the top. And then, so now, at this point, the active site histidines are restored to their former selves, right? We have a lone pair here and a proton here. And we also have that top piece of RNA, that sort of 5' end of the whole RNA molecule, is also now free to go. So at this point, RNase A has reset itself to where it started in terms of its active site. It's ready to catalyze another reaction. RNA is split in two, and we consumed one water molecule. And that's what you see in this little equation box here. So that brings us to our second mechanism for enzyme catalysis, which is covalent catalysis. And covalent catalysis can be defined as the formation of a covalent bond between the catalyst and the substrate, or a catalyst-substrate bond. And this usually happens because of nucleophilic catalysis, where a nucleophile on the enzyme attacks the substrate to form that covalent bond. And when we think back to our Bronsted-Lowry acid and base definitions, bases can be good nucleophiles. Maybe not because of the Bronsted-Lowry definition, but bases are often able to donate electrons, right? So that's what makes them good nucleophiles. However, In the course of making this covalent bond between the catalyst and the substrate, it's important to remember that that bond eventually has to be broken to release the product. And so the enzyme during this covalent catalysis substrate bond formation, it can't make the bond be too strong because that bond does have to get broken at the end. So let's look at an example of covalent catalysis. And this is the reaction which is decarboxylation of acetoacetate. So we see in this picture on the top the uncatalyzed version of this reaction where we have acetoacetate. We have this little carboxyl group on the end is electrons are attacking nucleophilically over at this carbonyl carbon, and then the electrons are being pushed up to that carbonyl oxygen, which ultimately helps to release this carboxyl late group as a carbon dioxide. But these the transition state essentially that formed is an enolate ion, right? And this is a really unstable ion. It has this oxyanion that really is trying to get rid of these electrons. And so if a proton can be taken up from the environment, that essentially can happen, right? Because then that oxyanion is able to push electrons back down towards that carbon to make the carbonyl group again, and then the double bond is pushed over to the right. And then if you add a proton here to make CH3, then this is a much more lower energy, more stable configuration of atoms that is called acetone, right? But this enolate sort of transitional state, right, is really, really unstable. And so, using an enzyme or catalysis to help do this can help you to make a lower energy transition state. And we can do this through covalent catalysis. So if you have an enzyme that has an amino group that's able to interact with the substrate, it can bond to make a shift base where the carbonyl was, right? And because of because now we have a nitrogen here that's bonded to that is making 4 covalent bonds, it has a positive charge, right? So when we had that same electron movement that we saw in electroacetate to lose this carboxylate ion, where the electrons on this oxygen are being pushed back over here, what the nitrogen, because it's positively charged, is much happier to accept that extra pair of electrons as a lone pair, because as we know, nitrogen is very happy to have 3 bonds and a lone pair of electrons. And so ultimately this helps, we have that same sort of reversal of our transition state where we have the electrons being pushed back out towards the right and the acceptance of a proton. And then the enzyme releases from to reform this carbonyl over here, right? And then we have our same product, which is still acetone. And so the reactant and the product in both cases are the same. What changed was the transition state, which the enzyme through covalent catalysis was able to make a lower energy transition state, which ultimately helps the reaction go faster. So let's talk about another type of catalysis mechanism, and this one's going to be metal ion catalysis. So metal ion catalysis is carried out by metalloenzymes, and these enzymes have tightly bound metal ion cofactors, and it can be one of a pretty substantial number of metals, including iron, copper, manganese, cobalt, magnesium, and zinc. And metal ions are helpful for catalyzing reactions because they can do a few things. They're able to bind to and orient the substrate in a particular direction. They are able to help carry out redox reactions. And they are also pretty good at stabilizing negative charges. So let's look at an example of metal ion catalysis. And this one is going to be our carbonic anhydrase enzyme. And so the reaction that it carries out, again, is shown here in the green box on the right. So we have carbon dioxide plus water makes a proton plus bicarbonate ion. And so if we look at the active site of our carbonic anhydrase, we have our zinc ion that's shown over here and in the middle, right? And it is surrounded by a number of different histidine residues. which each have an imidazole ring. So that's shown here in this diagram with IM for imidazole. So there's 3 different histidine residues, there's 3 different imidazole rings, and they're all helping to coordinate with this central zinc ion. And the zinc ion has one more bond that's available to form, right? And so it does this by actually interacting with a water that enters the active site, and it essentially forms this negatively, it takes that piece of a water molecule which loses a proton, and this particular group would be extremely unstable usually, right? It's got these extra electrons on it, it's negatively charged, but because it's interacting with the zinc ion, this helps to stabilize this oxyanion, while it is then able to help do this reaction with the carbon dioxide. So now we have all these extra electrons, they're attacking that carbonyl carbon, and ultimately helping to form this transition state that we see in the middle, that again, the zinc ion is helping to stabilize by coordinating with this oxygen. And at the same time now, you can already sort of see the shape of our bicarbonate molecule that is going to be the product of this reaction. And so then with the addition of a second water molecule, that helps to displace that bicarbonate ion product, and then we sort of reset the enzyme in its original state, where that water ion, the oxygen and the hydrogen are coordinating with that zinc ion, and then the proton is free. And then, of course, we have our product, which is the bicarb that we released. And so you can see how that metal ion, that zinc, helps to stabilize the reactants a lot to help make this reaction happen. So let's look at some other enzyme mechanisms. And so what we're going to talk about next are proximity and orientation effects. And so this essentially is 2 different things, right? Proximity means how close they are to each other, and orientation means which parts of them are next to or facing which other parts of this, meaning like the enzyme and the substrate have to be, or if there are multiple substrates, they might have to be, have particular parts of them in a certain orientation, right? And so, so for these types of effects, we have to have reactants that are both coming into contact with each other, proximity, and that are also oriented correctly. And so enzymes that help to increase proximity of reactions or reactants can result in a 5-fold increase in the reaction rate. Enzymes that help to orient the reactants with each other can help up to 100-fold increase in rate. And then enzymes that cause reduced flexibility of the substrate or the transition state can result in up to a 10 to the 7th-fold increase in rate. So each of these different factors can help make a reaction go much, much quicker. And so sort of similar things to what we were talking about before in terms of stabilizing the transition state, things like charged groups in that active site, as well as the exclusion of water, can help to facilitate the binding of the substrate, as well as to help stabilize that transition state. So let's talk about an example of proximity and orientation effects. We're not actually looking at a specific enzyme here, we're just looking at this SN2 reaction to help get an idea of what proximity and orientation can do to help. So we have our initial reaction at the top. So we have imidazole, which is shown in green, that is doing that indole pair on the nitrogen is doing a nucleophilic attack onto this carbonyl carbon in the paranitrophenyl acetate molecule that we're showing at the top. And we have this movement of electron that helps to basically make 2 different products. So the imidazole ring attaches to the carbonyl carbon, and we get this N-acetyl imidazoleum, excuse me, and this paranitrophenolate ion. So yeah, essentially we are making one bond and breaking another bond, right? And so this reaction is not super fast, but you can change it up a little bit by looking at this reaction that we have going on the bottom. So in the first reaction, our two reactants were separate and individual molecules. But if you bring them closer together in space by adding this linker to them, you do two things. One, you increase the proximity, right? They're physically closer to each other. And two, because of the size and the flexibility in the linker, we also help a little bit with the orientation so that nitrogen with the lone pairs is very close to this carbon that it's going to attack. And so ultimately this reaction on the bottom proceeds much more quickly than the reaction at the top because of proximity and orientation effects. And enzymes can do a similar thing where they'll bring substrates or reactants closer together, both in space and in orientation. So next, let's look at other mechanisms that enzymes use. And so now we're going to talk about preferential binding of the transition state. So what does this mean? So enzymes that preferentially bind to the transition state can first result in increased strain on the reactant, right? So if an enzyme is a really good fit to the transition state, it might be a little bit more work to get the reactant into that active site, but then once it's there, it's going to want to proceed really quickly to that transition state. And you can, and so because of that lowered, or that smaller change in energy required to essentially reach the transition state, because the transition state is being stabilized, this is going to help result in that lower activation energy. At the same time, because of that lower activation energy, it's going to result in an increased concentration of the transition state. And once that substrate has reached the transition state, the reaction could proceed in either direction, right? It's downhill both to go back to reactants as well as to proceed forward to products. But because overall that there's more transition state, you will get essentially more of both, right? We'll get more of what happens in either direction, as well as that increased rate of the reaction, where the reaction proceeds much faster. So overall, this preferentially binding of the transition state can make a reaction proceed more quickly. So let's look at what happens with the transition state and think about some inhibition properties of enzymes. So let's start with this example. So we have L-proline as our product, as our reactant that's getting made into D-proline, which is the product. And the enzyme that does this removes the one hydrogen from this chiral carbon in the center. From the back, the proline proceeds through this planar transition state in the middle, where the chiral carbon is essentially flat, or has 120-degree angles, and then the enzyme then puts the proton back on to make the carbon chiral again, and it does that from the front. So the overall configuration about this chiral carbon has changed. But the enzyme that does this is really good at stabilizing this planar transition state. And so if you ever wanted to make that enzyme not be able to catalyze this reaction of turning L-proline into D-proline, a really good way to inhibit that enzyme would be to use another type of molecule that looks a lot like the transition state, because this type of molecule is really, really good at binding the transition state. That's what we were talking about before, right? It's going to preferentially bind the transition state. So if you want to inhibit this molecule, you wouldn't necessarily start with a molecule that looks like the reactant. You would want to choose a molecule that looks like this planar transition state. And so that's why on the bottom here, we have two examples of inhibitors for this enzyme. And they are going to be more planar, right? Because of the structure of where the electrons are placed around the ring in the molecule, much more so than proline, right? So, so that's sort of an example both of how stabilizing the transition state can help a reaction go faster, and also can help you start thinking about good ways to inhibit particular enzymes that might be dependent on that enzyme's mechanism. So now that we've talked all about defining and categorizing enzymes, you are ready to answer the practice questions.